Hello, everyone. How are you? So we're having a new class. So we are done with mitosis already. So we'll be talking about the cell now. So the new topic we'll be discussing together is cytology, which is the study of cell. Um, you will notice that the way I've been passing it across, I'm trying to um communicate um, in a way that you can understand it, as if we are in the class, as if we are interacting, so that it will be more like um, I'm physical, like you can see me and I can see you. So I'm trying as much as possible to just do it that way. And I decided to make these slides so that I can also read out points. So as I'm going through it, you also can also do the same thing and understand what I am trying to say. And although very soon, we'll still try as much as possible to see how we can do some what's it called physical classes online, which I'm going to connect every one of you, and we can discuss and possibly have question and answer um period amidst ourselves. So all the same, like I said, just make sure you are serious with this, and believe me, you wouldn't have any problem answering your question during the examination period. Once we are done with every topic, go to the past question, extract all questions that has been asked, extract it, buy your 8 in 1, 10 in 1, and use it to start answering questions the way you want to answer it in the examination hall. So with that, you'll be practicing. So once you come across any question like that, you know what to do without stress. So cytology is the study of cell. Um, we've done embryology, we've done mitosis and meiosis, which is cell division. We've done microscope, and here now is the cell. So if you look at it, every of those topics are still around each other. So it makes you understand every of the topic very well. Let me just put it that way. And maybe possibly after this, I don't know if I should go to tissue next or I should start with genetics so that most of those terms that we mentioned here, which we also have them in genetics, so we'll be able to rotate around them there. But any which way, let's continue with this. So after this, the next topic you are going to see. So this is an example of a cell, animal cell precisely. That's what I decided to put in here. And the cell is made up of organelles. Organelles is just like when you have a plain paper or a plain sheet of paper, and you put gum on them, on it rather, you put gum on it, and you begin to place smaller particles, different object on it. That's more like the way it is looking at the diagram. So the smaller object that you put inside the cell, like I said, you have a paper and you put gum on it. Now that gum that you apply on every part is more like the cytoplasm. So now on the cytoplasm, you now have a lot of organelles, or within or inside the cytoplasm, then you now have a lot of organelles there, which are what we have here and well labeled. And I want you to take note of those labelings because it's very important. IGMB, JPEP, they do ask this question. They can just draw this diagram or something related to this and ask you to, they draw lines out of them and ask you to label them. So try as much as possible to look at every structure and know the labelings. And like I've always said from the beginning, look at all the first alphabets of the organelles and use it to make a word. Use it to make a word. Any word, the way any word you feel you will always remember. So that when you see cell, you know the word that make up the whole organelles, and it's really, really going to help you. So I will just mention, if you look at this, you see the mitochondrion, you see the plasma membrane, you see the cytoplasm, you see the microtubule, you see the centrosome, you see the microfilament, you see the lysosome, you see the smooth ER, endoplasmic reticulum, you see the vacuole, you see the secretory vesicle, the peroxisome, the Golgi vessel, the Golgi apparatus, the chromatin. I think we've talked about chromatin, we mentioned them. Nucleolus also, yeah, we mentioned it under, what's it called? Cell division. The nucleus itself, the rough ER, and the ribosomes and intermediate filaments. So some of those things are what we'll be discussing about. We might not really talk about every one of them, but as a good student, I expect you to just check other ones and try to know them. So it's just as simple as that. But the main ones, which most exam will want you to label, these are the ones we'll be discussing about. So the cell structure as seen under light microscope. Now we're trying to see how it looks like in animal and in plants, both left and right from what you can see here. In animal, the cell is bounded by a thin cell membrane. So the whole cell is bounded by a thin cell membrane, which is called the plasma membrane. You can see from the diagram we have here. And note, protoplasm consists of nucleus and cytoplasm. So I want you to take note of that. What is protoplasm? Protoplasm is what? The nucleus plus the cytoplasm. They make up the what? The protoplasm. Now, cytoplasm now contains the what? The organelles. I think I mentioned that from the beginning. So you have the idea of what we're talking about. So when we say cytokinesis, now you can remember what the cytoplasm is. It contains all the organelles, and that's that. And coming to implants, how does it look in plants? So in plants, cell is bounded by a thick cellulose cell wall. Now that's the difference now. Animal cell has cell membrane covering it, bonding it. Um, Plant cell has cellulose cell wall all around it. Now the center is taken up by a large vacuole. There's a very, very large vacuole in plants, but in animal, we have very, very small vacuole, as you can see from the diagram. They are small vacuoles, and a very good example is when you talk about food vacuole in amoeba, I think contractile vacuole also in amoeba. So all those give you what the vacuole is all about in animals. Now, nucleus is located along the peripheral of the cytoplasm outer edge. Now, look at the diagram here. Look at where your nucleus is situated at one side. Are you getting me? Outer edge. But in animal, it can actually be at the center. It can be anywhere. But basically, because of the large vacuole in plants, you can see the nucleus at one side. So the two different type of cells are, we have the prokaryotic cell and we have the eukaryotic cell. Now, the word pro, anytime you see the word pro means force. Anytime you see the word EU means pro. So a prokaryotic cell, karyotic means nucleus. So karyotic means nucleus. So it's more like saying a forced nucleus cell and a true nucleus cell. Now, a forced nucleus cell can be seen in bacteria and cyanobacteria. Under what's it called? Plant diversity. I know you must have come across this, and if time permits us, I'll be able to touch all those parts too under plant diversity. So, but let's just take it one after the other here, and let's just make sure we follow up so there wouldn't be any issue at all. Now, eukaryotic cell, the protists, the fungi, the plants, and the animals, they have the true nucleus. And what does it mean by having a true nucleus and a false nucleus? You notice that your true nucleus will be enclosed by the nuclear membrane. They will have nuclear membrane. But a false nucleus doesn't really have a nuclear membrane. What do I mean by that? It means the materials or the chromatin materials are scattered. You can see them almost everywhere in the cytoplasm. It's not as condensed or as surrounded like what you have inside the eukaryotic cell. Now, if you look at the diagram, this very diagram represents is a diagram for prokaryotic cell, how it looks like. You also have the cell membrane and the chromosome and the cell, as you can see. So this is trying to tell you that what it is just scattered inside the cytoplasm. Even in some diagrams, you just see it scattered, like it doesn't have a defined point where you can see them. But in eukaryotic, as you can see, everything is in one place. So that's that. And classification of eukaryotic cell. Now, so basically, we want to talk about that of eukaryotic cell, a true cell, a true nucleus cell. So, unicellular organism consists of only one cell. Example is amoeba, chlamydomonas, and euglena. They have only one cell, and with that one cell, they can perform all the activities of a living organism. Because when we talk about parasites of organisms, they may stand Niger card, possibly more than that then now. So yeah, amoeba can actually go through the whole thing. So with just one cell. Now, multicellular organisms consist of two or more cells, and we have a lot of example here: Hydra, man, Spirogyra, and so on and so forth. Now, the history of cell. So we won't just talk about some scientists that participated in talking about the cell or discovery of cell. And one of them is Robert Hooke, which so I would just want you to go through this. That is why it's in form of PowerPoint, so you can always pause it and write out everything you want to write out, so you'll be able to have idea about it. So he's an English scientist, and he actually discovered honeycomb and structure of a cell rather in 1665. So his book, which is Micrographia, and he said the cork is made up of components which he called the cell. So he saw a cork, and so he said that the component he noticed in this cork looks like something, and at the end of the day, he gave it the name cell. So Felix Dudassin, which is a French biologist, also discovered cell is made up of living component or substance, and called the living substance protoplasm. So it's more like everyone is just making discovery, and they give it a name. So if you remember the word protoplasm, I mentioned it in the previous slide. I said protoplasm is made up of what? So like I said it's made up of the nucleus and the cytoplasm. The nucleus and the cytoplasm make up the word the protoplasm. So when you're talking about the protoplasm, the word Felix comes to mind. Matthias Sheldon, a German botanist, in 1983 revealed that the body of plants is also made up of cells. So he also contributed that, okay, fine, you people are talking about animal, even plants has what? Cell. So that is Matthias coming out from there. And Theodor Schwein, which is a German zoologist, 1839, discovered body of animals made up of cell. So Sheldon and Swan discoveries led to the postulation of the cell theory. So through Theodore, we're able to come across the cell theories, which we'll talk about soon. And Rudolf also, which is a German biologist in 1855. So he concluded, sorry, please. So he concluded in his research that all cells come from pre-existing ones. So what does that mean? It means new cells come from old cells. Let me just put it that way. Meaning there will always be a cell before there will be another cell. And that explains or leads to mitosis and meiosis. You understand? Because in mitosis, you have a cell going through multiple cell division to produce 2 new cells, and 2 new cells again produce 4 new cells, 4 produce, and so on and so forth. So pre-existing cells, which means old cells come from the what? Sorry, new cells come from the what? From the old cells. So the cell theory that we made mention of is a cell is the structural and functional unit of life. So that's more like the general definition for cell, which we all know. So we now say all living organisms are made up of cell. So when you talk about living organism, I mention living organism, you should know that they are made up of what? Of cell. Okay, I might need to minimize this so you'll be able to see it properly. Okay. All cells come from pre-existing ones. So all cells now come from pre-existing ones, which is one of those things we mentioned earlier. And there's no life apart from the life of cells. So when you talk about life or no life, so every life, when it is living, rather, you definitely talk about what? The cell. So all living things are either single cell, which is unicellular, or group of cells, which is what? Multicellular. And the next one, the important cell structures and functions. So out of um, the cell structures we now have now, the important ones, we want to try to talk about them. And like I said, it's not like every one of them are not important, but based on what our syllabus want us to lay emphasis on, that is what we are trying to do here. So let me just place this here for now. When it's time, I will remove it and bring it out. So the cell wall, we talked about the cell wall earlier. We said cell walls are found in plants. So definitely everything we'll be talking about here will be more of what? Of plants, because animals don't have cell wall. Now the primary wall, which is the primary wall of a plant, during cell division consists of cellulose fibrils. This run through a matrix of other complex polysaccharide. It may later thicken to become secondary cell. Now we're trying to like, okay, we have primary cells, which comes up during cell division. Yeah. So when they become thickened and they now have complex polysaccharides, they become thickened and then they becomes the secondary cell. Now all that is just trying to tell us that what those are the secondary cells now make it very strong. When you talk about trees, so in trees now you have the secondary cells making giving it a kind of strength, flexibility. Are you getting me? So that wind cannot just break it or anyhow. So that is one of the functions of cell wall. We'll get there and we'll still talk about the function. Now the matrix consists of polysaccharides divided into pectin, and the middle lamella consists of thin layer pectin also substance, which is made up of calcium and magnesium. Now, if you are talking about a bone, which also help in support in human being, so in animals or human being generally, so you always make mention of some minerals, and these same minerals now are performing the same function, doing the same function in plant also, which is calcium and what? And magnesium. So they help neighboring cell walls, so bring everything together and strengthen it, so preventing form of breakage or making it more flexible. Okay, so that's that. I think I can make this smaller here. I hope so. No. Okay. So the next point, plasma consists of fine pore in the cell wall. The pore is lined with cell surface membrane. Now, all this is just trying to explain how the cell wall, what the cell wall is made up of. So the term plasmodesmata rather, try to take note of it because these are some of those questions that they ask. Are you getting me? In cell wall, they say write short notes on cell wall. Write short notes on cell wall. So these are some of the points that you just need to just put together just give you a short note. So plasmodesma encourages a continuous system of cytoplasm. That's the simplest to be formed between ring. That also tells you one of the function of cell wall. So transportation of substances within the cell also. So that's the function of plasmodesma, which is found inside the wall, inside the cell wall. If you look at the diagram actually shows some of those things that we talk about. The middle lamella here, the primary cell wall, which later become thickened later through the help of polysaccharides to form the secondary cell. So we have it here. Plasma membrane, they also have plasma membrane. These are the cross-linking glycan. Glycan is a type of polypeptide that's what's it called, carbohydrates. This is also pectin here. As you can see, the network of transportation of materials linking things, linking things together, and also for strengthening of the whole cell wall. So now functions of cell walls, which we have here. One, it provides mechanical support and protection. I think I've talked about that earlier also. There will be mechanical support and protection of the whole plant. It prevents, so which means it helps to regulate what's it called, osmotic pressure inside the plant, minerals inside the plant. It helps to regulate, so it will not be it will not absorb too much of water. Are you getting me? So there will be balancing of nutrient and water going in and out. So it's a pathway for movement of mineral salt and water. Yeah, that's the pathway. We once talk about it in the other part. So it allows a pressure potential to be developed, which supports. So, like I said, um, the mechanical support talking about the pressure that it's built up within itself, to preventing any form of leakage and all those stuff. So it has specialized function through various modifications, such as what lignification, when they have lignin. Also, still talking about what mechanical support for strength of the plant. So the next one is the cell membrane. So in animal cell, it forms the cell boundary. So as we have in what's it called, plants, where we have the cell wall forming the outermost part, which is more like the boundary. So in animal here now, we have the cell membrane forming the cell boundary, and it's partially permeable. Partially permeable, so which means it's not all substances that can enter through the cell membrane, but there are some that are allowed. Example, water, gas, glucose, fatty acid, all these ones that fuse slowly. So it is made up of protein and lipids. And one of the reasons why they will always ask you to take protein, because of your cell membrane also. So it's proteinous substance rather. So because of the cell membrane, and definitely protein helps in what's it called, bodybuilding. So you always have protein and lipid almost everywhere, because the layers of the skin, for example, the outermost part is made up of lipids, and that's fat and oil, lipids basically. And you also have protein there. Now, phosphorus molecule consists of bipolar edge, that's phosphate, and two non-polar hydrocarbon tail from fatty acids. So, still talking about lipids, which fat and oil and all stuff that have been explained. So from Daniel and Davidson model, which says that the center core of membrane composes of double-layered phospholipids, and the outer layer composes of monomolecular protein layer. Now, all Daniel and Davidson model, all they are trying to say, they are trying to let us know what make up the cell membrane. Are you getting me? So the phospholipid And the outer layer, which we have the protein layer, which is basically found in the cell membrane. What are the functions of cell membrane? They regulate in and out movement of substance. So it's not all substances that will go in and go out. And those that are even going in and going out, they are being regulated. So it's not just going in and going out. There are regulations to them. They link adjacent cells together by membrane junction. So when you have one cell, remember, we are more like talking about a multicellular organism. They are adjacent cells, so they link them together so that in multicellular organism, they will all function as one. They can transport materials from one cell to another. They act as receptor sites for recognizing hormones, neurotransmitters, and other chemicals. Now, the cell membrane, like I said, is more like the outer part of your body here. That's just an example where you have your what's it called, neurotransmitters and all the stuff. So information are being passed from one cell to the other through the cell membrane. That's all we are just trying to talk about there. And number three is the nucleus. We are very familiar with the nucleus, and I think we've discussed so much about the nucleus. So when you talk about the nucleus, the true nucleus, you talk about in eukaryotic cell. So let's just go through what we have here. So it's found in eukaryotic except for phloem C tube element and the mature red blood cells of mammals. So what we are trying to say is we are trying to talk about where we cannot see the nucleus. The frame sieve group element, which is mature, you will not see there, and also in mature red blood cells. Now, paramecium has two nuclei. Yeah, from unicellular organism, I think you would have come across that. So the micro and the meganucleus. The skeletal muscle contain multiple nuclei, the skeletal muscles. The nuclei are conspicuous because they are the largest cell organelles. That's the nuclei. I'm talking about plural now. They are conspicuous because they are the largest cell organelles. Now, I want us to what's it called? Um, take note of something. The reason why I'm putting it in form of points here is when people want to say, okay, write short note on nucleus. Are you getting me? So just put it out in form of points like this, so it will be easy for an examiner or someone who is marking to look at it and say, wow, this is point. Mark whatever you want to mark and go. Instead of writing stories and maybe the first 2 points are not making any sense to the person just angrily, you understand, mark the whole thing wrong. But if you put it out in form of points like this, it makes it easy for any examiner to see what you are trying to pass across, and immediately they grant you what you are supposed to have. So skeletal muscle, like I said, it has multiple nuclei, meaning more than one nucleus when we talk about skeletal um, um, muscle cells. So it's enclosed by a nuclear membrane. Yeah, so the nucleus is enclosed by a nuclear membrane, which means two membranes, nuclear membrane, two membranes, perforated by nuclear pore. So, which automatically means there are pores on the nuclear membrane or the nuclear envelope. And this pore also allow only some materials to go in and out, not all materials. So they also change barrier between the nucleus and the cytoplasm. Now, the outer membrane is continuous with the endoplasmic reticulum. Let me just go back. I want you to just see what I'm trying to talk about. Now, if you look at this diagram now, this is the nucleus now, and this is the nuclear membrane surrounding it. This is the nuclear membrane. Now, the nuclear membrane has a pore. This is an example of a pore. So you just see some pore here. And the continuation of the nuclear membrane now form your what? ER, endoplasmic reticulum, either rough or smooth, um, depending on where they are. So nuclear pore allow exchange of material between endoplasmic reticulum. So there is exchange of material between the nucleus and the cytoplasm there. Now, in nuclear DNA, plus protein form a fine network of thread called chromatin. Now, we've mentioned chromatin also while we were discussing mitosis and meiosis. So when DNA come together with protein, we call it what? Chromatin. Now, during cell division, chromatin threads become chromosome. So that is why I said most of the things we mentioned while we were discussing um, cell division, also we will still talk about them here because there is a link between them. We've talked about chromatin material, I'll call it chromatin thread. They are the one that later form what? The chromosome. Now, nucleolus, it is a round, darkly stained structure inside the nucleus. Its function basically is to make ribosome. So where are ribosomes? You can see the word in the nucleolus. Are you getting me? And the nucleus is found inside the word the nucleus. So the stain occur due to large amount of DNA and RNA in it. So the dark color or stain that we said they have is due to the large amount of DNA and RNA that we can see in them. What is the main function of nucleus? It controls the cell activities because it contains the genetic information in the form of DNA, which is an organized gene. So that's that. The main function is control the cell activities, controls all the cell's activities. Okay, and go to the next one here, which is the cytoplasm. So it's the jelly-like transparent, slightly viscous aqueous fluid composed mainly of water. So we have large amount of water in the cytoplasm. Like I said, cytoplasm is just like the gum that you put on top of what's it called, a sheet of paper before you now begin to place other organelles on it. So that's just what it is. So that's why it's jelly-like and transparent, like this aqueous fluid. And many organic and inorganic substances dissolve in it. Yeah, because there are a lot of things that you put in them. So either organic or inorganic substances, a lot of them dissolve in heat. All metabolic process occur inside the cytoplasm. Like almost every other organelles are placed inside the cytoplasm. So in cytoplasm, there are many ultracellular components known as inclusions and organelles. No, so since I think I've been talking more of saying they have organelles, they have organelles. So not just only organelles that they also have what we call what the inclusions. Inclusions are temporary structure which are mainly starch, grains, oil globules, and excretory crystals. So when you say inclusion, they are temporary structure. So they are not going to stay there for a long time. So that is why we don't really mention them like that. Are you getting me? So starch, grains, oil droplets or globulets, excretory crystals, and so on and so forth. So you will see them there. So they are temporary structures. So inclusion. What is inclusion in the cell? You should be able to tell us temporary structure. And organelles are now the permanent structure and distinct part of a cell which has a particular structure and function. So organelles, all other structure apart from inclusion found inside the nucleus is said to find inside the cell is said to be what what's it called, organelles. And even endoplasmic reticulum, the nucleus itself, we assume it's organelles, the Golgi, and so on and so forth. Now, examples of organelles are ER, endoplasmic reticulum, Golgi apparatus, centrioles, and so on and so forth. Just give me a few minutes, please. Thank you. So, as I was saying, sorry for that, just quickly need to attend to something. So, those are the examples that we've given here: ER, the Golgi apparatus, centrioles, ribosomes, and so on and so forth. So, those are some of the examples of organelles. Almost everything we are talking about here, they are organelles. So, ER, endoplasmic reticulum, is the most extensive cytoplasm. So ER also is a type of cytoplasm, but it's very extensive organelle in the network of membrane that forms the ER. So the membrane encloses a space that is continuous throughout the network, three-dimensional network of sac-like and tubular cavities. Now, the ER consists of flattered membrane-bound sac called the cisternae, forming tubes and sheets. It is continuous with the outer membrane of the nuclear envelope. So we talked about it under nucleus. So we mentioned ER there. So it's a continuous one. And basically, you see them. ER may be covered with ribosomes to form the rough ER, or ribosomes will be absent, form the smooth ER. So we have two types of endoplasmic reticulum, ER, which is the rough and the smooth. If it is rough, it means it has ribosomes. If it is not rough, it means it does not have what? Ribosomes. So what are the functions? Rough ER is concerned with the transport of protein made by ribosomes on its surface. That's rough ER. So they transport those protein that the ribosomes on them make out. That's through the cisternae. Smooth ER is site of lipid and steroid synthesis. So just take note of that. That gives a difference between rough ER and the smooth ER. The rough ER basically, again, it will transport the protein that is made by the ribosomes on them. But the smooth ER, they are the sites for lipid and steroid synthesis. Okay, the ribosomes here now. So we'll talk about the ribosomes since we mentioned them under the rough ER. So ribosomes, they are more round that are freely in the cytoplasm or bonded to the rough ER. On ribosomes, protein molecules are synthesized from amino acid using genetic information carried by RNA from DNA in the nucleus. So now there are type of RNA. We have messenger RNA, we have ribosomal RNA, and we have the transport RNA. So now you said on the protein molecules are synthesized from the amino acid using genetic information carried by the RNA. Now this RNA now, which is the messenger RNA. Messenger RNA will leave nucleus and come out of the nucleus. Now, the ribosomes inside the ribosomes here now, which are protein molecules, we now synthesize amino acids that will have the same what's it called? The same um base as the messenger RNA. Okay, I think when we get to what's it called? What's this topic called? I think it's going to be the next topic after this. So I'll talk about that. Messenger RNA, DNA and RNA. Yeah, we'll talk more on them. So you'll be able to know the differences between them and how they function. So, like I said, these are the proteins, the amino acids that are used by messenger RNA to form DNA. So ribosomes are small organelles that consist of large and small subunits. They are made of roughly equal part of protein and RNA. So made up of those equal parts, protein and RNA. So the slightly smaller ones are found in mitochondria. So if you are looking at the smaller ribosomes, you will see them inside the mitochondria and the chloroplast. So functions now, they are sites of protein synthesis, basically. Ribosomes are sites for what? Protein. So when you talk about protein, when you talk about amino acid, remember amino acid is a, what's it called? It's a subunit of protein. So when you talk about protein synthesis, you come to the ribosomes. The seventh one, the centrioles. They are rod-like structure found near the nucleus. Rod-like structure found near the nucleus. They are at kind of right angle to each other in animal cell. And what do? Cell division. They help in cell division. While we're talking about mitosis and meiosis, you remember very well, we talk about the centrioles. The centrioles, which produce spindle, fiber, which helps in cell division. Golgi apparatus, which was first discovered by Camillo Golgi in 1898. So using the special staining techniques, and it consists of stack of flattened membrane bond sac called the cystinae, together with the system-associated vesicle, that's small sacs called the Golgi vesicles. Now in the diagram, okay, let me just show you that again, in case many of us do not look at it or see it properly. Okay. So you will see it here, the Golgi vesicles, you can see them here, and these are the Golgi apparatus. Okay, so okay, good. So what is its function? I said if it's form okay, I've talked about that. It transport materials to the other part of the cell or the cell surface membrane for what? Secretion. That is its function, basically. It helps in the processing of many cell material in the system, e.g., proteins from ER. And lastly, secretion or formation of lysosomes. So just take note of the functions, their functions. So transportation of materials, that's from the cell to the surface, to the cell surface for secretion. We talk about processing of cell materials in the system, protein from ER, getting protein from endoplasmic reticulum, the Golgi apparatus help with that. And lastly, it helps in secretion or formation of what? Lysosomes. So that takes us to lysosomes. What are lysosomes? It's a special spherical sac bonded by a single membrane. It contains digestive enzymes like proteases, nucleases, and lipases. Now, what are we? If you hear the word proteases, meaning something related to protein, nucleases, something related to nucleus, and lipases, something related to what? Lipase or lipids. Now, these enzymes, what do? They break down protein, one, nucleic acid, two, and lipids, respectively. So lysosomes contain these enzymes to help break down protein, nucleic acid, and lipids. So the enzymes work best in acidic medium stroke environment. So enzymes in lysosomes are synthesized on rough ER and transported to the Golgi apparatus. Remember, lysosome also are found, what's it called, secreted from the Golgi apparatus, secretion of lysosomes. So here now, we now said what? The, okay, the enzyme work best in this, okay, yeah. Enzyme in lysosomes are synthesized on ER, rough ER, and is transported to that Golgi apparatus. So the Golgi vesicles containing the process enzyme later bud off to form the lysosomes. So that's how lysosomes are being produced. So they'll be budding off from the Golgi vesicles. So then you have them as a lysosome. Therefore, lysosomes arise from the edges of Golgi body. Larger secondary lysosomes are formed by fusion of primary lysosomes. The smaller lysosomes form the larger lysosomes with small vacuole. So vacuoles arise by the infolding of the plasma membrane. So the plasma membrane, when there is infolding of plasma membrane, then you have what? The vacuole. If you remember food vacuole in amoeba, how it is being formed, it is by the infolding the infolding, it fold inwardly. Infolding of the plasma membrane will give you your the vacuole. Now, primary lysosome may also fuse with antophagosomes. The primary lysosome may also fuse with what? Antophagosomes. That's a small membranous pocket enclosing worn-out organelles. Antophagosomes. So it's a small membrane, pocket-like, which encloses worn-out organelles. So organelles that are worn out, that are not more functioning, they engulf it. They engulf it, just like amoeba going for its food material, and it engulfs it. So once it engulfs it, it now begins to digest and break it down before it is being sent out of the body. So antophagosomes, but okay, including worn-out organelles like the mitochondria and ribosomes. So lysosomes play important role in the various cells that make up the defense system of the body. So lysosomes also help in defending the body, because all worn-out tissues, organisms that are not, or organelles, or materials that are not good again for the body, they enclose it and find way of sending it out of the body. Functions: digestion of material taken in by endocytosis, bulk transport of materials through membranes of the cell. Now, you can just notice that when you talk about maybe this is it boy that they call it, that after some time you press it, something comes out. Yeah, that's one of the ways. That's one of the function of lysosomes. Now, sending out in form of small vesicle or vacuole materials that are dead in the cell, send it out and you can place it out, put it out from your body. So digestion of materials taken in by endocytosis, endo, inside, bulk transport of materials through membranes of the cell. And endocytosis occur by infolding or extension of the cell surface membrane to form a vesicle or a vacuole. So the same thing, it just fold and come out in form of pimples or all those stuff. So that's an example of what the lysosomes can do. So we have endocytosis is of two types. Endocytosis is of what? Two types. Endo, internally. So phagocytosis, cell eating. The materials taken up is solid in phago. Phago, eating of cell, materials that are solid. Pinocytosis, drinking of cell, that's cell drinking. So the materials taken in is what? Is in liquid form. So please try to know the difference between phagocytosis here now and pinocytosis. Phago is drinking of cell, and pino is what? Sorry, phagocytosis is eating of cell. Pinocytosis is drinking of cell. Now, how you can also differentiate it? And when you talk about phagocytes, when you talk about phago, like eating, phago deals with solid material. Pino deals with what? Liquid material. So thank you very much. And okay, another one is here, autophagy. This is the process by which unwanted structure within the cells are engulfed and digested within lysosome. It will now release, it's a release of enzyme out of the cell, that is exo. Remember, endo is different from exo. Sent out. Waste material removed cell, e.g. undigested remain from phagocytic vacuole, or useful materials may be what? Secreted. So if you look at all those things that we make mention of now, we make mention of phagocytosis. We should know the difference between phagocytosis, endocytosis, which is made up of this and that. So phagocytosis, pinocytosis, and coming to the next one, which is autophagy and exocytosis. So let's try to go through those what's it called? Those terms again, those words again, so that we'll be able to differentiate them one after the other in case we see them as short answer question. Microbodies, a roughly spherical organelle bonded by a single membrane. They are spherical organelles, so they are bonded by a single membrane, not like the nucleus and I think mitochondria that have double what's it called, bonded membrane. So this one is just single membrane. It contains, it appear finely granular, except for occasional striking crystalloid of filamentous deposits. What is the main function? They contain catalase, which is an enzyme that breaks down hydrogen peroxide. They are associated with oxidation reaction. So oxidation reaction in the cell is the work of microbodies. And also, they contain catalase, which is an enzyme that breaks down hydrogen peroxide. In plants, they are sites of the glyoxylate cycle. Glyoxylate cycle, okay, that's another topic, a full topic under respiration, something. So we have more to talk about that too. So microbodies are very important also from as what's it called, an organelle in the cell. The next one is mitochondria. The next one is mitochondria. They are surrounded by an envelope of two membranes. Outer is smooth, and the inner membrane is folded into sheets or tubules called matrix. The matrix contains few ribosomes, a circular DNA molecule, and phosphate granular. A circular DNA molecule and phosphate granular. That's the matrix, what it contains. They are called the powerhouse of the cell. So mitochondrion is the powerhouse of the cell, while nucleus is dead to control the activities of the cell. So what's the function of mitochondria? They help in aerobic respiration. So when you talk about respiration, you will definitely have to talk about what's it called? Mitochondria. So aerobic respiration, respiration that involves air. The cristae are sites of oxidative phosphorylation and electron transport, and the matrix in the site of Krebs cycle enzymes. So all this, when we get to respiration under plant, under animal. So we'll talk more about this aerobic respiration and also anaerobic respiration. But just remember, one of the functions of mitosis is it helps in aerobic respiration. Chloroplast, from the word chloroplast. So you know chloroplast is found in plants, and these are colored plastids containing the main photosynthetic pigment, which is called chlorophyll. So you can see the chlorophyll inside the chloroplast. It is found mostly in the mesophyll cell of leaf. The mesophyll cell of leaf, that is where you will see the chloroplast, and it is surrounded by an envelope of two membranes. So they too, chloroplast also has two membranes, just like the mitochondrion. The outer smooth, the inner membrane is thrown into lamellated structure, showing two distinct regions. So when you talk about the mesophyll cell, you talk about the chloroplast, which is the chlorophyll, which is the main photosynthetic pigment found in chloroplast. So we now say here that there are two distinct regions when you're talking about chloroplast. So we have the grana of the chloroplast and the stroma of the chloroplast. The grana, or gramma rather, oh, the stroma, let's start from the stroma. The stroma forms the matrix of the chloroplast. The stroma forms the matrix of the chloroplast. It is the site for the dark reaction of photosynthesis. Remember, in photosynthesis, we have the light reaction and we have the dark reaction. So the stroma basically help in okay, we'll go soon. Sorry for that. So the stroma forms the matrix of the chloroplast, and it is the site where we have dark reaction of photosynthesis. So it contains ribosomes, cellular DNA molecules, lipids, droplets, starch, starch grains also. It contains that. So the grana now is site for light reaction. So that is where light reaction occur. That is the grana of the chloroplast. Now photosynthetic unit known as the quantosomes are located on the grana of chloroplast. We'll still talk more about this also when we get to photosynthesis in plants. So we'll talk more on grana of the chloroplast, talk about the more stroma of the chloroplast, light reaction, under light reaction and dark reaction in photosynthesis. We'll still talk more about that so you'll be able to understand more. But basically, what is the function? It is the organelles in which photosynthesis take place. So sugar is produced from carbon dioxide and water using light energy. So light energy is now converted into chemical energy, and the chloroplast contains chlorophyll and carotenoid pigment. Number 13, the vacuole. So as we earlier said, we said the vacuole actually takes a lot of space in plants, but in animals we have vacuoles being small. We have small vacuoles compared to what we have in plants. So it's a fluid-filled sac bonded by a single membrane, tonoplast. So tonoplast is a single membrane that surrounds the vacuole, and tonoplast is partially permeable. So animal cells contain relatively small vacuoles, which I've talked about, such as the phagocytic vacuole, the food vacuole, the contractile vacuole, the autophagic vacuole. Now, the phagocytic, the autophagic, we also talk about them also. When you talk about phagocytes, so vacuole. So all this help in what engulfing this what's it called? Digestion, that's for the food vacuole, for example, in unicellular organism, digestion, the contractile vacuole for breaking of undigested waste, mainly liquid in liquid form and so on and so forth. Like the one we have in paramecium. So plant cell, notably mature parenchymal cells, have a large central vacuole. The fluid they contain is called the cell sap. So the vacuole contain a fluid which is called the cell sap. And this cell sap is made up of mineral salts, sugar, organic acid, carbon dioxide, oxygen, pigments, enzymes, waste, secondary product of metabolism, and so on and so forth. So, which automatically means because it has all this, it is going to be concentrated. So it's going to be concentrated, and that makes us also be able to talk about osmosis, diffusion, and all those stuffs in between the cell, how they absorb materials from the soil through what's it called, through the root to the stem and to every other part of the plant. So function, it stores various substances which includes waste products. That's one of it. Sometimes plant vacuole contain hydrolytic enzyme which function or act as lysosomes. So it makes an important contribution to osmotic properties of the cell, yeah, which I mentioned, osmosis. So under what's it called? Photosynthesis and all those stuff. So we'll still talk about that, which is plant diversity also. So we'll still talk more about that forms and function, plant forms and function, talk more about it. And water generally enters the concentrated cell sap by osmosis through semi-permeable tonoplast. You remember what the tonoplast is, which is the single layer of the vacuole. So vacuoles sometimes contain pigments in solution, so which include anthocyanin, which is red, blue, purple, and other related compounds, shades of yellow. With us, those are the colors you can see in flowers, in roses, and everything. So that's about the vacuole and its function. So what are the differences between plant and animal cell? So plant cell has vacuoles, and animal cell, we would definitely say they have small vacuole or absent. So you can say plant cell has large vacuoles, but in animal, it is small or it is totally absent. Plastid, or plant cell has plastid, chloroplast, chlorophyll, are all present in the plant, but we don't have all this in animal. The cellulose cell wall is found only in plants. Cellulose cell wall, you can't find it in animal. Um, Plant stores carbohydrate as starch granules. That is the way they store it. But animals, they store carbohydrate as glycogen. Now, plants store lipids, but animals store it as what? As fat. So that's our development. Centroles are absent rather in plants, but in animal they are present. Protoplasm is less dense. Protoplasm is less dense in plant cell, but has more dense protoplasm in animal. So what are the similarities? They both possess, meaning they both have the cell membrane, they have the nucleus, they have the cytoplasm, they have the ribosomes, Golgi apparatus, and so on and so forth. So those are the things that um, similarly. So some of the questions here, they said, explain the terms functions of the cell membrane. Functions of the cell membranes. So, but please, anytime they ask this question, basically you always see it in number 1 or number 2 in your exam. That's the theory part. Anytime they ask, try as much as possible to get up to 10 points, just point 1, point 2, make it up to 10. So that if it is just going to carry 5 marks, already you even have more than 5 points there. And if you cannot write up to 10 points, just make sure your point is more than 5, because most cases, 5 points, 5 marks is being allocated. So that ends what's it called? The cell generally. But please, I will still want you to do more of your work. Like I said, try as much as possible to go to your past question, check all the questions that has been asked under the cell. Under the cell, check all questions that have been asked and answer everything. Once you answer everything, there's something about this A-level program. They don't have new questions. They come back with old questions. And funny enough, you can even sit down and predict the question that's going to come out. It is very possible you can predict questions that will come out. If you have all your past questions, start from 2000, for example, start from 2000 till date. Okay, they asked this year. Most cases, when they ask this year, for example, they will skip 2 years before they ask again, skip 3 years before they ask again, skip 1 year. So if you sit down and you do the calculation, you will predict the questions that will be coming out during your own set. And that makes it so interesting for students that can sit down and that all sense. So thank you very much. God bless you. We'll see in the next class. Like I said, if you have questions, just write it and give it to your coordinator to get to me. Then we'll know what to do about it. Thank you very, very much. Thank you very much. Really appreciate you guys.